Okay, today's topic is going to be growth and metabolism. We're going to be looking at metabolism, but metabolism is what drives all growth. So it will be a discussion of the chemical reactions that take place within plants that cause them to make their own food and cause them to be able to grow. And we have to think about this as an energy flow. And that's basically what is going to be going on here. We're going to take energy from sunlight and we're going to convert that energy into usable chemical energy that the plant can use to grow and that we can use as food for our own bodies and as food for the animals that we keep and for all the other organisms that consume plants for food. And we start with a discussion of photosynthesis. Photosynthesis can be described as the reaction of life. Now, we can have life on this planet without photosynthesis, and we did at one time billions of years ago. But the planet was a very different place at that time. The atmosphere was completely different. Everything was completely different. And when photosynthesis as a process evolved, it changed everything, primarily due to the generation of oxygen in the atmosphere that allowed for other higher forms of life to evolve following the evolution of the photosynthetic pathway. So it is indeed the most important reaction on Earth because all of our food depends upon photosynthesis. And not only does all of our food depend upon photosynthesis, but the oxygen that we breathe also depends upon photosynthesis. Without photosynthesis, we would not have much on this planet. And you also have to think about photosynthesis as a means of extracting carbon dioxide from the atmosphere, because that's exactly what we're doing here. This figure here is an estimate, of course, 5.4 to 8.1 times 10 to the ninth kilograms, or 1 to 1.8 million tons of carbon is fixed. And by fixed, we mean changed from CO2, atmospheric carbon dioxide, fixed into sugars and other organic materials such as starches through the process of photosynthesis. So we're taking quite a bit of carbon out of the atmosphere through the photosynthetic process. And we do know that carbon in the atmosphere is one of the major pollutants that we have on this planet now, leading to global warming and all sorts of other problems. But one way of removing some of that carbon, and this will be sort of a theme throughout, is that we can take it out through photosynthesis. And agriculture itself is a system for exploiting photosynthesis. Everything that we grow depends upon this reaction, so we want to exploit this reaction in order to produce corn and wheat and all the other crops that we grow. The overall photosynthetic reaction looks something like this. And this is a balanced equation, and this is not exactly the way things happen, but at least it's in an overall viewpoint. Essentially what we're doing here is we take 6 molecules of carbon dioxide, we combine that with 12 molecules of water, and with light as an energy input, and in the presence of chlorophyll, which is a molecule that is designed to capture light energy, with a series of reactions. We have a long series of reactions here that will lead to the production of glucose, C6H12O2. That should be O6, by the way, C6H12O6, plus 6 oxygen molecules, plus 6 water molecules. So you see, it's sort of funny that we go from 12 to 6 here. Well, you could put 6 in here and balance the equation without this one over here. You could do that, but that's not exactly what is happening. So we have to represent this as much as possible as the way it is actually happening in the various stages that go on in here with these intermediate reactions. And again, this is a summary. It's like, it's only showing the beginning products and the end products. It's like saying steel plus rubber in the presence of labor and a bunch of other machinery and other things gives you an automobile. So it's about the same level of representation there. So let's look at some of the individual steps. Basically, we divide photosynthesis into two separate series of reactions. One series of reaction is called the light reactions, and of course the other series of reactions would probably be called the dark reactions. We haven't gotten there yet, but we will on the next slide. Basically, we call these the light reactions because these are the reactions that harvest the energy of sunlight. They use sunlight energy directly and convert it to a usable chemical forms. Now, we're not going to convert light energy into sugars at this point. That comes next. But what we do is we convert light energy into very unstable chemical compounds that can be used later, some unstable high energy compounds that can be used later to make the more stable glucose and other sugars. So what happens is that the chlorophyll will absorb the light energy. Of course, you would have to have light to make these reactions go, and that's why they're called the light reactions. They absorb light energy. They raise the electrons in the chlorophyll molecule. Chlorophyll is a molecule that is a very complex molecule, and it has a cloud of electrons around it, and these electrons are raised to a higher energy state by the absorption of this light energy. And then when those electrons fall back down into their lower energy state, they re-emit that energy, and that energy then can be captured along a chain of several intermediate molecules. This is a process called electron transfer, resulting in the production of two things: ATP and NADPH2 along the way. These compounds, remember these compounds, these are going to be your energy-rich, unstable compounds. And of course, these two compounds occur in all living cells at all times. They are the energy currency of the cell. An ATP molecule, as you'll see, is identified down here, spelled out, adenosine triphosphate. That's where we get the ATP. And then NADPH is nicotinamide adenine dinucleotide phosphate, NADPH2. Again, you notice a couple of things here with these molecules. One of them, they contain the word phosphate, both of them, so that we know that phosphorus is involved here. Phosphorus is one of the nutrients that we'll study later on, and this is one of the functions of phosphorus, is it functions in energy transfer. So basically what you're doing here, you've got to sum this up, you've got, and I will sum it up on another slide, but you have chlorophyll absorbing energy, passing it along through a series of intermediate molecules, and then winding up with these energy-rich compounds. And these energy-rich compounds, ATP and NADPH2, can be used to drive other reactions down the road to create sugars. Again, this process is referred to as photophosphorylation. Looking at a chlorophyll molecule, and here's a couple that I got off the internet here. Basically what you have here is a ring structure. You notice that in the center of this ring structure is a magnesium molecule here, and over here on this side as well, magnesium. And that magnesium molecule is surrounded on all four sides by nitrogen molecules. Here, 4 of them. So you have magnesium and nitrogen, and then surrounding those are these carbon ring structures here. There are some double bonds in here. There are some various radicals of different types that represent different types of chlorophyll molecules, and you can have you have long tails on some of these things, and there are different branch molecules here that represent different types of chlorophyll. Chlorophyll A, chlorophyll B, and you notice that the red part here is this part of the molecule, this outlined in red here, is referred to as a porphyrin ring. Now, porphyrin rings are fairly common among living organisms. In fact, we share a common bond with the chlorophyll molecule in our blood, because hemoglobin, the hemoglobin molecule, is also a porphyrin ring. The only difference between, or the major difference between chlorophyll and hemoglobin is that in hemoglobin you have here, instead of a magnesium molecule, you have an iron molecule, and that's why blood is red, because of the presence of iron. Chlorophyll is green because of the presence of magnesium. So another thing we need to recognize here, that this molecule contains 2 other of our important nutrients, nitrogen and magnesium. So you would expect that anytime you have a deficiency of either one of these two nutrients, you're going to suffer. Your chlorophyll content is going to be lower, your photosynthesis is going to be less efficient, and you probably don't have the nice deep green color that you would have if you have plenty of these nutrients there. And in fact, one of the commonest nutrient deficiencies that we see is chlorosis, or yellowing of the leaf. And if we can often correct that by adding nitrogen. And we also know that when we add nitrogen to plants, we generally decrease or increase the deepness of the green color. So one of the ways of making your lawn or a golf course or an athletic field look deep green is to add nitrogen to it. And again, that helps with the manufacture of the chlorophyll molecules. So these electrons, again, these electrons in the highlighted regions here are the ones that get excited. There's a cloud of electrons here. If you studied any organic chemistry, you understand how these things work. But there's a cloud of shared electrons among all these carbon molecules that surround this. Each place there's a joint here. There's a carbon molecule that's not shown. For example, this is a carbon, this is a carbon, this is a carbon. So carbon makes up the backbone of this stuff. And these things share a cloud of electrons. These electrons become excited, they rise up to a higher energy state, and then eventually that energy will decay and the electrons will fall back down. This is caused by absorption of light energy. A photon hits this chlorophyll molecule, it increases the energy level of its electrons. When they fall back down and release that energy, it is captured in the form of intermediate metabolites, eventually leading to the production of ATP and NADPH2. So again, it's about energy flow. Light energy converted into chemical energy that we can use and that plants can use to grow. And then we have the dark reactions. This is also referred to as the Calvin cycle, commonly called the Calvin cycle, after the fellow who discovered it. Now, the dark reactions do not use light directly, but they use the products of the light reactions, and they cannot necessarily occur in the dark, because if you have darkness, then you don't have the light reactions, so you don't have the dark reactions going either. But they're called the dark reactions because they don't need light directly as an input. They use the inputs of the light reactions, but they do nonetheless, make no mistake about it, they do need to take place in the presence of light. If you supply cells, for example, with adequate ATP and NADPH2, the products of the light reactions, then they can conduct the dark reactions in the dark, but they don't normally have that in nature. The dark reaction goes like this. And here's where carbon dioxide comes into the equation. It doesn't enter into the light reactions at all. That is something else. Here you take CO2, carbon dioxide, from the atmosphere, and that's the only place you can get carbon in a plant is from the atmosphere. You add to it NADPH2, an energy-rich compound, ATP, another energy-rich compound, and through a series of intermediates you will release water, and you'll have a carbon chain. This carbon chain will eventually, of course, become a sugar later on down the road. NADPH2 supplies the energy. Remember, it got the energy from the light reactions, and it supplies protons in the form of H+. ATP, again, supplies energy. The carbon chain here, this can either be a C3 carbon chain or a C4. This chain that results as that comes about as a result of this can either be 3 carbons long with different molecules, or it can be 4 carbons long. There are 2 different metabolic pathways that are used by plants, not the same plants, different species of plants. Species of plants will use one or the other of these 2 different pathways, but not both. Certain types of plants are C4 plants, certain types of plants are C3 plants. Sugar formation goes like this. You take your carbon chain here, either the 3 carbon chain or the 4 carbon chain. You add more energy in the form of ATP, and again a series of reactions will result in the production of glucose, C6H12O6. That is the ultimate product of photosynthesis. This will generally be converted into sucrose for transport within the plant, and then eventually converted into starch for long-term storage if that's what is needed by the plant. Again, here ATP supplies the energy, getting that from the light reactions. So overall, what we have here is essentially energy input. Remember, this is all about energy. Energy coming from sunlight into the light reactions with the input of water and the release of carbon dioxide. This is where the carbon dioxide comes off. You have to have ADP and NADP plus. These compounds will be converted eventually into, through the light reactions. These are the building blocks. Adenosine diphosphate. Basically all that happens here is that molecule is converted into adenosine triphosphate, so that another phosphate group is added to that molecule, and this is a very high energy bond. And when that bond is broken here in the light dependent reactions, they call them here, what I was referring to as the dark reactions, they call them the light dependent reactions. This is also, there's an enzyme involved here called rubisco that we'll talk about in just a minute. CO2 comes in, water comes in, you get a end product here that is glucose, and this is the C3 cycle that we're showing here. So basically everything is sort of in a cycle here. This generates, once these ATPs and NADPH molecules are stripped of their energy, they convert back to ADP and NADP and go back and are fed back into the light reactions over here. So they're cycled throughout the process. And again, the main things that are given off here are going to be your oxygen here, your inputs, or your oxygen here. Here's your oxygen, your CO2 in, or water in, CO2 comes in here, up here in the light reactions, and again water is given off. So water is used as well as given off, and that's the reason why it's written the way it is in the overall reaction that we had at the beginning. One important thing I would point out here is to remember the name of this enzyme here. This is the enzyme that starts everything. This is the enzyme that grabs CO2 out of the atmosphere and starts this process, rubisco. I'll give you a fuller name for that in just a minute. So some of the factors that affect the rate of photosynthesis. First of all, why are we interested in the rate of photosynthesis? Well, since agriculture is a system of exploiting photosynthesis, we are very interested in the rate of photosynthesis because we want photosynthesis to occur at its maximum rate at all times if possible. Of course, that's not possible. There are many things that influence the rate of photosynthesis, and we have some of them that we can manage and some of them that we have under our control as individual producers and farmers, and some of them we cannot. For example, one of the things that is important here is the concentration of CO2 in the atmosphere and within the leaf, the concentration of it, how much CO2 there is there. Inside the plant leaf, it's regulated by the stomata, or the little holes in the leaf that allow gases to pass in and out. And the openings and closings of these stomata are primarily influenced by water status of the plant. If the plant has good water status, in other words, there's plenty of water available, those stomates will be open, those stomata will be open, and gases will be being exchanged. As the water status in the plant deteriorates, in other words, there's not enough water in the soil or wherever the plant is growing to keep the plant going, those stomata will close to prevent further water loss from the leaf. And when they do that, they decrease the amount of CO2 that can get in also. It's sort of a trade-off. The plant has to regulate the amount of water going out through the stomata, and in doing so, it also automatically regulates the amount of CO2 coming in. Now we know that greenhouse and growth chamber studies have shown increased photosynthetic rates as a result of increased concentration of CO2. As your CO2 concentrations go up, photosynthetic rates go up. But we can't really do that as a manager unless we want to invest in a greenhouse that is pure CO2, and plants won't grow in pure CO2 either. They also need oxygen. Normally, CO2 concentration in the atmosphere is about 360 parts per million. This has increased from about 250 parts per million in 1850. So we've seen maybe a 50 or 60 or 70% increase. Actually, that's about 40% increase in CO2 concentration since about 1850, primarily due to the release of fossil fuels, burning of fossil fuels, and other things that emit large amounts of CO2. Another thing another factor that influences photosynthetic rate, not unrelated to the one we just discussed here, is water. Water is a very important, even though less than 1% of the water absorbed by the plant actually enters into the photosynthetic reaction. It does show a lot of water, 12 molecules going in, 6 molecules being released, but that's less than 1%. Most of the rest of that water is used to fill the cells, to keep the plant in good water status, and to keep the stomata open. The stomata have to be open for CO2 to get in and out. You have to have water to keep those stomata open. When you decrease the amount of water, the stomata close so that the plant can maintain its water status. And this is one thing we can do something about as growers and producers. We can irrigate. We can manage water in other ways. Most people think of management of water strictly in terms of irrigation, but there are lots of other things you can do to help make water use more efficient by the plant, and we'll discuss some of those a little bit later on when we talk about water. A lot of them are soil-related factors. Some other things that influence photosynthetic rate are going to be light. How much light? If you don't have light, you don't have photosynthesis. Normally we're going to measure light in terms of intensity, duration, how long it is, and frequency, or another word for frequency that we often would use would be more commonly color. What color is the light that we have? So, and intensity, of course, is brightness. So brightness, how long it shines, and what color it is. These are the three ways that we measure light. Each one of those will have some impact on photosynthetic efficiency. Currently, only about 4 times 10 to the minus 10th. That's what that should be, 10 to the minus 10th here. A very small amount of the sun's energy reaches Earth. The rest of it just radiates out into space or hits the other planets. Or this would amount to about 4.5 million horsepower per square mile, which is quite a bit of energy actually. Plants can only utilize about 1 to 3% of this, depending upon their individual rates of photosynthesis and what their water status is and what type of plant they are. Of the sun's energy, only about 44% of it is visible to us. These would be the light frequencies that range from 390, a very high frequency, resulting in a violet color, to 760, lower frequency, or far red, so the nanometers wavelength. So you have 390 to 760, and everything in between is going to represent different colors. You go from violet to blue to green to yellow, just like in the prism. And eventually you wind up with the lowest frequency, the far red light at the 760 nanometer wavelength. Chlorophyll works mostly to absorb light in the blue and red frequency range. It does not absorb just any color. And you'll see noticeably here that one color that it does not absorb is green. It reflects the green light. It does not absorb it, it reflects it. That's why we perceive to our eyes a green color when we look at plants, because they are reflecting the green, they are absorbing the blue and red of sunlight. And so this is what you get if you look at these are the wavelengths. Remember I said they range from 390 up to 760. So these are the common wavelengths in between, starting with the purples and blues over here, going through the greens and then the yellows, and then the reds over here on the far right hand side. And this is for a wheat plant. And if you look at the percentage of each one of these frequencies that it is absorbed, we absorb almost all of the red light, about half of the blue light, and very little of the green light. They can measure this through the laboratory. So we get a fair amount of absorption here, very little absorption here, mostly reflected, and then here we're absorbing most of this red light. So chlorophyll really likes red light in this far red category here, around 700 nanometer wavelength. And we have to look also at intensity. So we've looked at color. Intensity is also another important factor here in light, how bright the light is. And we can measure sunlight intensity from 0 to 100%, and we can measure photosynthetic rate here as a function of sunlight intensity. And what we see is that with a plant like tobacco, at about 50% intensity, we are at our maximum photosynthetic rate. So that any additional brightness doesn't really increase photosynthetic rate at all. We see a similar thing here slightly to the right here for soybean and wheat. These plants do not become, this is what you call light saturation. These plants light saturate at fairly low intensity levels. They light saturate. On the other hand, plants like corn and sorghum will continue to increase photosynthetic rate as light intensity increases. These plants do not light saturate. And if we look at the metabolic differences between these, we will discover that these types of plants are your C3s, and that corn and sorghum and other types of plants are indeed C4. It's a different type of metabolism that results in these differences in response to light intensity. So we see that C3 plants reach the light saturation point, and this would be the light saturation point, the point at which any increases in sunlight intensity do not result in increases in photosynthetic rate. These light saturate at these relatively low intensities, less than 60% in most cases. And how do we measure photosynthetic rate? What kind of experiments can we perform to differentiate between a plant that has a low photosynthetic rate and one that has a high photosynthetic rate? Well, all we have to do is grow these plants in a chamber where we can measure the rates of change from CO2 to oxygen. And plants that are converting CO2 into oxygen at higher levels, of course, would have higher photosynthetic rates. It's very easy. We have instruments that can very easily measure the concentration of CO2 and oxygen in a closed system with a great deal of accuracy. Now, temperature is also a factor. And temperature, again, will affect C3 plants more than it will C4 plants. What we see is, if we look again at photosynthetic rate here on the y-axis, measuring the differences in CO2 absorbed versus O2 released, and we look here at temperatures within a constant light intensity, temperatures decreasing from low to high, and we don't necessarily say what temperatures we have here. We're trying to measure these temperatures within those that plants would normally grow. In other words, we're not going to have 200 degrees Fahrenheit on this scale, because you wouldn't be having any plant living at that. We're probably going to be running these, depending upon what species they are, from maybe around 50 degrees Fahrenheit up to about maybe 110 degrees Fahrenheit. These are reasonable temperatures for plant growth to occur. And what we see is that C3 plants will increase their photosynthetic rate as temperature rises to a point. And then at some point, the photosynthetic rate starts to drop off at very high temperatures. Again, for C4 plants like corn and sorghum, that photosynthetic rate will continue to increase as the temperature increases, and it really doesn't maximize. Of course, it'll get to a point where any higher temperatures, everything drops off. But within normal operating temperatures, you will get continual increase in photosynthetic rate in C4 plants, more so than you do C3 plants. Now, the other things that in fact that influence photosynthetic rate is carbohydrate translocation. That is when you make sugars and you make starches. And these sugars and starches need to be moved out of the way. They need to be moved to wherever they need to be absorbed in whatever part of the plant they need to be in. If there's a backup in that, in other words, you don't have demand for carbohydrates anymore, you will get a lower rate of photosynthesis. So this source, whatever the source is, the source, remember we have a sink and a source. It is actually the sink demands. Sink demands influence photosynthetic rate, how much your sink is demanding of the carbohydrate. As that demand lessens, your photosynthetic rate will begin to go down. And of course, leaf age is also an important factor here. As things get older, as leaves get older, they tend to become less efficient. Now let me go back just a minute here. I wanted to say one or two more things about this business of C3 versus C4 plants at temperatures. A part of this is due to differences in anatomy, internal leaf anatomy. The C4 metabolism pathway is a later evolving pathway. It came about later. Everything used to be C3. And the common question that you might ask here is, if C4 plants are so much better than C3 plants, why don't we just use C4 plants in agriculture? And the reason for that is because there are certain environments on this planet, most environments, as a matter of fact, that are just not conducive to growing C4 plants, like corn or sorghum or millet or other types of C4 plants. The fact of the matter is that C3 plants like wheat and soybean are better in certain places and are more productive in certain areas, particularly those C3 plants that are cool season plants. And most cool season plants, those are plants that are planted in the fall and harvested in the spring, like wheat, grow better at lower temperatures in general, and therefore they're not really affected by this difference here, by this high temperature, and they grow with less water. And that's one of the key things, is you can grow wheat during the winter without irrigating it. And this is commonly done in the Great Plains of the United States and Canada and in Asia and in China and India and Russia and other places where they have lots of land but not a lot of water resources. And you could not grow summer crops in some of these areas very well. You need these C3 plants to be able to exploit that environment. The other thing I would say is that one of the biggest differences here is between differences in this rubisco enzyme and its affinity for CO2. This thing has an affinity for CO2. It grabs CO2 out of the atmosphere, and as temperature goes up, the affinity for this changes, and it may not like CO2 as much at high temperatures in these C3 plants. C4 plants have a mechanism, an internal mechanism whereby they can concentrate CO2 within the leaf, and that's one of the things that makes them more efficient and less subject to this phenomenon of temperature, high temperature saturation, and then light saturation. So how do we manage plants to increase the efficiency of the photosynthetic reaction? This is one of the key things about this class, is to tell you how to take advantage of photosynthesis and how to make it more efficient. One of the things that you can do is you can increase your leaf area index over time by decreasing your row spacings or distance between plants and increasing the plant population again, which decreases the distance between plants. LAI, remember, stands for leaf area index. It is the leaf area over the land area. And of course, when you plant a crop, your leaf area index, remember, LAI needs to be between 2.5 to 4, but it starts out at planting at 0. There is no leaf area index. So you want to get from 0 to here as quickly as possible. And you have a timeline here, certainly. And at some point after emergence, you're going to have a leaf area of index of 0.1, and that will begin to increase and eventually get to where you're trying to be. But the idea is to shorten this time span as much as possible. So you can do that by planting your plants closer together, decreasing row spacing or increasing plant population. There were some classic studies conducted in the 1960s by a couple of guys at Iowa State University by the name of Scheibels and Weber. Scheibels was a plant physiologist there, and Weber was their soybean breeder. And they performed some very simple experiments on soybean where they looked at dry matter accumulation. And let's talk for a minute just about dry matter accumulation or dry matter production. Dry matter, of course, is the part of the plant that doesn't contain water. So we can measure the dry matter in any plant at any time we want to simply by cutting that plant, putting it in an oven, and drying it for a couple of days, and weighing what is left, driving off all the water and weighing what is left. And that's exactly what they did in this experiment. And of course, your dry matter accumulation is going to be a function of photosynthetic rate. If your photosynthetic rate is functioning and your photosynthetic efficiency is high, you'll get greater dry matter accumulation. If your photosynthetic efficiency is low, you'll get less dry matter accumulation. They discovered that in this case, dry matter accumulation was a linear function of solar radiation interception. So they measured, what they did was they measured dry matter accumulation and solar interception and found a linear relationship. As the amount of sunlight intercepted by the leaves increased, and they could measure these types of things, then your dry matter accumulation over time increased. There was a definite linear relationship there, a very straight line relationship, not a curvilinear relationship, but a straight line relationship. And in fact, if they planted their soybeans on 40-inch rows, 40 inches between rows, they would have row spacings here, and they would put 40 inches between rows, a fairly wide row spacing. This would be 40 inches. Say this is 40 inches here, 40. They would have less interception, less total interception, and would have delayed time to maximum solar interception compared to row spacings where they planted, and if we diagrammed it over here and assume again we have a 40-inch row spacing, they would plant these on 20-inch rows, put an extra row in the middle there. These rows would tend to close up, close this canopy, close this leaf canopy a lot faster because the plants and the rows are closer together. So they found that indeed, if they planted plants in this pattern, that the light intercepted would grow at a greater rate, and it would take less time to get to this desirable leaf area index at that row space, at that closer row spacing. It would seem today that this would be a fairly obvious thing, but of course at that time nobody had actually done the research to show this, and this is what Scheibels and Weber did. We can also vary things like the planting direction, the direction of our rows. Generally north-south rows are going to be better than east-west rows simply because the sun is coming up over here, and you got the sun coming up, and you'll get better coverage than if you have the sun coming up over here. Let's say the sun is setting, it's the same difference. You have a lot of sunlight falling between the rows, more so than you would at the east-west direction. Plant breeding, leaf position and architecture. It's better to have leaves that position themselves for maximum solar interception, and we can do this through plant breeding. Planting date also has an impact. When we plant early. Plant as early as we can, such that we get to maximum leaf area index at an early date as possible. Of course, we have certain restraints with planting dates. We can't just go out there on January the 1st and start planting and say we're planting early. Sure, we can plant early, but plants will die if they're warm season crops. And even if they're cool season crops, they won't make it either if you plant in January. If we maximize by planting date what I mean by that is to plant as early as possible within the recommended range of planting dates. And then, of course, if we provide the plants with adequate moisture and nutrients, don't let these things be limiting factors. Make sure the plants are well fertilized, make sure they have as much water as you can get them, either through irrigation or other water management strategies that we'll discuss later. And then, of course, pest control, pest management. Keep the diseases away, keep the insects away. A lot of what these pests will do, insects, for example, they will eat the leaves. And if they're eating the leaves, you're taking away your photosynthetic apparatus. Diseases will do the same thing. Diseases will cause various problems with leaves. They'll cause leaves to become chlorotic. They'll cause leaves to become necrotic. Tissues will die. You'll start to get lesions on leaves, and when you have this, you're reducing your photosynthetic efficiency. Now, the other metabolic reaction that we need to look at here is respiration. Basically, plants have to breathe just like humans do, just like animals do. And while animals do not carry on photosynthesis, we respire. Plants respire and carry on photosynthesis. So respiration is the process of obtaining energy, getting the energy back out of the sugar molecules, or the starch molecules, or the other organic molecules that are produced as a result of photosynthesis. Respiration occurs at all times in all living cells. Photosynthesis only occurs in those cells that are exposed to sunlight in plants. Respiration is needed in plant cells to provide energy for division, cell division, growth. Basically, it's what we're talking about here. Cell division and elongation is the way plants grow. And then the other metabolic processes that are used to produce energy for the synthesis of oils, for example, fatty acids, fibers in cotton and other plant types, protein in soybean, etc. All the other different metabolic plant parts that we use. There are basically three types of respiration. You have your aerobic respiration, your anaerobic respiration, and photorespiration. And this is the one that really we need to just focus on just a little more than the other two. Aerobic respiration occurs in the presence of oxygen. This is the common type of respiration that we see in animals. You take a glucose molecule, C6 there, take a glucose molecule, you convert it back into carbon dioxide, water. Here's this other 6 molecules of water that we lost before, and the release of 40 ATP molecules, which we can then use to drive these other reactions. Again, ATP is the energy currency of the cell, adenosine triphosphate. Essentially, this is the opposite of photosynthesis. It doesn't require light. It's referred to as the Krebs cycle. It occurs in an organelle in the cell called the mitochondrion, or plural mitochondria. So basically what you have here is sort of a diagram here that shows a lot of terms and a lot of things, and basically it's a cycle. There's a lot of different things going on here, but the main thing you need to remember from this is the fact that this is the overall reaction here, the breakdown of sugar into the release of water, carbon dioxide, and ATP. Aerobic respiration occurs when oxygen is not present. There's not sufficient oxygen, and cells can undergo aerobic respiration temporarily. This occurs in the cytoplasm. It's also referred to as glycolysis. Sugar molecule be converted into ethanol plus carbon dioxide plus water plus 2 ATP. So you can see this is a much less efficient type of photosynthesis, 40 ATP molecules versus 2 ATP molecules. But we don't want to do this unless oxygen is not sufficient. And that happens occasionally. You know, that's why they call it anaerobic respiration without oxygen, but you can have such a thing as anaerobic exercise. This means that you're exercising at such a level that your body is not able to use oxygen entirely to release all that energy that you're expending. It has to rely some on this, and then it'll have to go back up and make this up later. But this can also lead to a production of a product called lactic acid. This is another type. This is what occurs in muscles But of course, this process, as shown here, is the main way that the main method of making alcohol for human consumption, brewing. Yeast can conduct this reaction quite well, and when you do anaerobic respiration with yeast, you release ethanol, which builds up to a certain level and eventually kills the yeast, and there you have something with a lot of ethanol in it, like beer. Lactic acid is another product, and this is what happens when you make silage or forage that is cut and fermented and produces lactic acid. That also will kill the organisms that's conducting the anaerobic respiration, and serves to preserve by lowering the pH, preserves the silage and preserves the grass. Now, photorespiration is another thing here. Again, it's called photorespiration because it occurs in chloroplasts in the presence of light. It's usually considered to be detrimental. It results in the oxidation of sugars just produced by photosynthesis. So here you have a cell that is photosynthesizing, producing sugars, and all of a sudden that sugar is being produced is turned right back around and converted right back into CO2 before it can be utilized as an energy source by the plant. Photorespiration is also dependent upon the concentration of carbon dioxide, concentration of oxygen, the light, having light there, and a temperature. It's a temperature related thing. The higher the temperature, for C3 plants at least, the more photorespiration is likely to occur. And again, it occurs because of this temperature dependent. It's a temperature dependent oxygen affinity of this rubisco enzyme. The rubisco enzyme, I told you, was a very important enzyme. It is the enzyme that starts the reaction of photosynthesis. The full name of this thing is ribulose biphosphate carboxylase oxygenase. And you see this hyphen here and these two suffixes, carboxylase and oxygenase. What this means is, this is why this particular enzyme will either grab a CO2 and convert it into sugar, that's why it's called a carboxylase, or it will grab an oxygen and cause respiration to occur. So it can function also as an oxygenase. The relative affinity for these two is temperature dependent. As the temperature goes up, it becomes more of an oxygenase and less of a carboxylase. And as your CO2 concentration goes down, it becomes more of an oxygenase. So all these different factors can increase the amount of photorespiration. Now, we don't see much photorespiration in C4 plants. Corn has very low rates of photorespiration. Other C4 plants the same way. And by the way, as a side note, most C4 plants are tropical in origin. They come from places that are known to have high temperatures. A lot of them are mostly monocots. There's only one species of dicot that is C4, and that is the species of Amaranthus, or pigweed. If you're a farmer or have anything to do with agriculture, you might recognize pigweed or Amaranthus or Palmer amaranth. It goes by lots of different names. There's various species of it. It's a very pernicious weed. It's very tough to control, and it's a C4 plant. But most of our other C4 plants are going to be monocots. Things like bananas, for example. Things that originated in these tropical countries, tropical places on Earth. Why do we have C3 plants in the first place? Well, one of the reasons for this and why we have so many C3 species, even though they have this so-called disadvantage, is because when photosynthesis as a process first evolved, there was no or very little oxygen in the atmosphere. And so these plants did not have to contend with oxygen as a competitor for CO2. So the C4 pathway came along later as an adjustment to this, but C3 plants still thrive on this planet because in most cases the temperature range that they're living in, the photorespiration does not become a burden to them. Only at high temperatures. When temperature gets hot, things get hot, photorespiration tends to increase, and you lower your photosynthetic rate or overall photosynthetic rate as a result of that. So photorespiration causes us to have to sort of recalibrate things, that and respiration, a normal respiration. If we look at here, this is our curve for photosynthesis as a function of percent sunlight, and we'll have respiration occurring at some fixed rate, generally a fixed rate regardless of what the sunlight intensity is. So you have this meeting here, this point here in the center where these two lines cross. This is referred to as your compensation point. At this point, the rate of photosynthesis and the rate of respiration cancel each other out. So there is a net carbon exchange of 0. As your photosynthetic rate continues to increase, you begin to see more carbon being fixed in the form of sugars than is being used. And this is referred to as the net photosynthetic rate. This is the amount of photosynthesis that occurs above what is being used, the photosynthate being used during the process of respiration. So the two important points here are your compensation point and your net photosynthetic rate. Net photosynthetic rate being photosynthesis, net here, photosynthesis minus respiration. And this is one of the reasons why we have this temperature dependent. As your temperature goes up, your photorespiration increases, and it would cause this to rise and give you a lower net photosynthetic rate. Other processes, just briefly, your transpiration. Of course, transpiration is the evaporative water loss from the leaves through the stomatal openings. This is again going to be affected by such things as temperature, humidity, and wind speed. As your temperature goes up, plants tend to transpire more. As your humidity goes down, plants will transpire more because of the greater ability for the water to evaporate from the leaf. And of course, wind speed. As wind speeds blow, as the wind is blowing, it will remove more water from the leaf. This water movement through the xylem, coming up from the roots, is called the transpiration stream. And we know that mineral ions, the nutrients that plants use, calcium, nitrogen, phosphate, potassium, these are absorbed and carried through the transpiration stream. So they're just following this thing, the water flow from the roots up through the upper parts of the plants. And transpiration serves to keep the plant cool, just as we stay cool by perspiration. You can have high temperatures, for example, but low humidity, and the plants will do just fine. Here in the south, we have high temperatures and high humidity. It can be very stressful for plants. But if you go to Arizona and irrigate plants out there at the very low humidities, they can tolerate temperatures of 105 degrees and be growing and be maintaining a fairly cool canopy, simply because there's plenty of transpiration occurring from the leaves. And then last but not least is the process of translocation. This is the movement of sugars, of sugars that are the product of photosynthesis. And other materials are transported, but it's mostly sugars, and they're transported again through the phloem. This is the other vascular tissue other than the xylem. This process is called translocation. So you have transpiration is water, translocation is sugar, and it is mostly sugar that is transported or translocated. Glucose, the main product of photosynthesis, is going to be converted into sucrose, and sucrose is nothing more than a disaccharide, or it's a glucose molecule joined together with a fructose molecule, two similar types of sugar molecules. They are joined together into a disaccharide and then transported throughout the plant, and then they may be used for whatever is needed at the time, or stored. When you're eating a watermelon and it tastes sweet, you're tasting sucrose. When you look at corn or sugar cane or some of these other plants from where we get sweeteners, most of that is a sucrose. So Where is the carbon on our planet? This is a question that we should all ask, since carbon is such an important part of our of our environment. Carbon dioxide is such an important part of our environment. If you look at the atmosphere, and this is the carbon that is in the atmosphere, that's in plants and soil biomass, we're looking at 2.1 times 10 to the 15 kilograms. If we look at the soil, the mineral fraction, this is the non-biological part of soils, we're a little bit higher, 3.5 times 10 to the 15 kilograms. If you look at dissolved CO2 in the oceans, and the oceans can have some, and do have some capacity to dissolve CO2 from the atmosphere. That's about a little more. 38 is quite a bit of it. 38 times 10 to the 15th kilograms. So the oceans themselves can absorb some of the extra CO2 that we're emitting, but of course that changes the chemistry of the ocean. It causes the pH to go down, for one thing, and some of the ocean plant life does not like to have those low pHs as much. But if you look at what's down below, fossil fuels, that's coal, oil, natural gas, carbonate materials, calcium carbonate, limestone, for example, these types of things on down below the soil layer, 43,000 times 10 to the 15th kilogram. So there's a lot of carbon locked up in our planet, way more than occurs in the atmosphere and the soil and in the oceans. But we do have to be concerned about this amount of carbon right here. So that's the end of the section on growth and metabolism. We'll be looking at flowering and fruits, I believe, next.